The next kind of trait that we come to are the multiple alleles. This is a trait where there are more than just two alleles or more than two options that can be inherited. So instead of just having a dominant and a recessive allele, we're going to see there are more. Now this is going to end up creating more phenotypes and more possible genotypes. In humans, blood typing is one of these. Your blood type is determined by which proteins are on the outside of the blood cells. There are 3 different genes that end up coding for your blood type. The first one is going to be I, big A, and this is going to code for protein A to be on the outside of the blood cell. So this is type A. I, big B, is going to be type B, and little I is type O. Type O has absolutely no proteins outside of it. The reason why I used I superscript A, I superscript B, and little I is so that the I's show us that we are using the exact same trait for all of these. The uppercase I's here and here show that A and B are dominant over O. The fact that both A and B are capitalized and both the A and the B are capitalized, that's going to show us that these are incompletely dominant with each other. And if we have a heterozygous form of that, we would end up with type AB blood. So let's take a look at the different genotypes that code for each phenotype. We have I, big A, I, big A, and I, big A, little I, that would code for type A blood. We have I, big B, I, big B, comma, I, big B, and little I, and both of those would code for type B blood. Then, as I was saying, A and B are codominant. So when they are together, we see a combination or type AB blood. In order to have type O blood, you need to have 2 little i's. Once you can understand this key, you will be more than fine working through your Punnett squares, because the Punnett squares work exactly in the same way as all of the other ones that we've worked with. So in this practice problem, we have a heterozygous type A person. That means they have to have type A blood, so they have to be in this category, but they have to have 2 different alleles. So they would have that genotype. That's going to be crossed with someone with type B blood who's also heterozygous. The second person has to come from this grouping and have 2 separate alleles. When we plug that into our Punnett square, we're going to see, we'll put our I, big A, little I, big B, little I, big A, I, big B, I, big A, little I, big B, little I, little I, little I. We come up with a lot of different genotypes. For our genotypic ratio, we are going to see that it has 1 I, big A, I, big B, to 1 I, big A, little I, to 1 I, big B, little I, to 1 little I, little I. Phenotypically, we are going to see that there are lots of options as well. That would be 1 type AB person to 1 type A, this would represent type A, to 1 type B, from here to 1 type O. That one with the 2 recessives shows type O blood. Hopefully this explains how multiple alleles work.